One of the reason nga nung again kailangan nga tanan nga part or tanan nga sample ang atong collect kay tungod if there is incomplete collection kay again ma-alter ang result. So these are the following. If ang first part or ang first portion kay ang imong ma-miss out, so it will result to decrease nga sperm count, increase nga pH, and specimen will not liquefy. Now, if ang last portion kay ang ma-miss out, kay it will give us a decrease semen volume, mo increase ang sperm count, mo decrease pH, and ang specimen will not clot. So, again, kung diri nga part kay uh, if ang first part kay, di ba, ang first part kay mo gina nag-consist sa bulk or kanang muna ang kinadaghanan nga part or amount sa ato nga semen. So if ang first part mo miss out kay mugamay gyud imong sperm count, ang mo increase yung pH kay tungod wala nimo na appeal ang prostate fluid, mostly sa prostate fluid nga again ang prostate fluid kay mo na siya ang naghatag acidity sa ato ang semen. So if ma-miss out nimo na siya nga part, mo increase imong pH, mo turn siya into alkaline. So ang imong specimen kay dili mo liquefy. Now if ang last portion ang imong ma-miss out kay mo decrease ang imong semen nga volume, mo increase ang imong sperm count, and ang imong nga pH kay sani mo taas o mo turn siya into acidic kay wala nimo na appeal mostly ang katong sa bulbourethral nga fluid. And again, ang bulbourethral fluid ka-responsible na siya for neutralizing or making our semen uh, uh, capable of kanang mabuhi pa ang sperm when introduced into the vagina of the females or in the vaginal fluid. So, mahitabo sad kay ang specimen will not clot. So again, we should take note of the following kay I think one ani kinigawa sa among board exam. So katong unsa either one of this kay unsa daw ang result if ang last portion kay imo nga ma-missed. So kana dapat gyud nimo nga kuan, dapat gyud nato ni ma-remember and dapat ma-take note nato what will happen if incomplete ang ato ang pag-collect sa ato ang sample. Now, for specimen handling, our semen is a potential reservoir for viruses like HIV and HBV. So, standard precautions must be observed at all times, and specimens are considered as biohazards. And for our semen analysis, so this will be the bulk nga part sa ato nga discussion. So for this part, we'll be talking about its appearance, liquefaction, or kanang again after diba after coagulation kay mo liquefy siya. So we ato sa i-discuss kung important na siya nga storyaan. Also for the volume and the viscosity. So ang consistency o ang gidaghanon sa ato nga sample for the pH and specific gravity. Kani nga part kani silang duha kay this should be the computation one. So ang sperm concentration, and then after na kay sperm count. Then we have our sperm motility, sperm morphology, and sperm vitality or viability. So now let's proceed to its appearance. Ang excuse me, ang normal nga appearance sa semen kay gray-white yang color, or other pod kay other books kay ilang term ani kay pearly white nga color, and it is translucent, and it has a characteristic of a musty odor, or other books sad or other nga mga scientist will define it as a Clorox nga order or kanang Zonrox nga odor, sorry. So again, musty odor o Clorox or kanang Zonrox type nga odor. So kana kabalo ko nga some of you kay kabalo gyud sa odor sa semen. So dili na ni siya bago sa inyo ha. So again, ang normal nga color kay gray white or pearly white. Now for its appearance, if ang we need to take note if the sperm concentration is very low. And we also take note that if increase ang iyang white turbidity, it means nga there is an increased number of leukocytes. And if there are increased number of leukocytes, meaning there is an infection. And also, we need to dapat lahi ang imong pag-count sa sperm cells, lahi imong pag-identify sa immature nga sperm cells rather og sa leukocyte og sa spermatids. Like, dili nimo sila pwede usahon. And also, if there is red coloration, meaning kay naay mga erythrocytes, and if there is yellow coloration, kay increase urine concentration, and urine is toxic to our sample. Okay, it will alter the integrity. Like, dili na siya motil ang sperm cell ana or sani dili na siya viable or dili na siya alive, dili na siya buhi. And also, one of the things nga mag-contribute sa yellow coloration kay some medications and prolonged abstinence. So, for liquefaction, ako ka sa picture. For liquefaction, kay fresh specimens are clotted. Analysis cannot begin until liquefaction has occurred. Kay if wala gani ni liquefy, dili ta ka-check sa iyang concentration, dili na to maka-count, dili ta maka-check sa iyang motility, dili na to ma-check ang morphology, at the same time kay dili na to ma-check if viable ba or buhi pa ba ang ato ang or ang mga sperm cells. So mo na nga dapat important jud kayo nga mo liquefy ang ato nga sample. And normal nga liquefaction time kay 30 to 60 minutes after collection. If fail gani siya nga mo liquefy within 1 hour or 60 minutes, it means nga ang patient kay naay prostatic deficiency kay tungod dili siya capable nga makapaliquefy sa sample. Now, if mula po na gani 2 hours nya wala pa gyapon ni liquefy, muna na nga mag nata i-add nga dulbecose phosphate buffered uh, saline equal volume sa kung pila ang volume sa ato ang sample, or mag-introduce ka og proteolytic enzymes like the alpha chymotrypsin or bromelain para lang mo liquefy gyud ang ato ang sample. So this two or these three rather kay mo ni sila ang i-introduce nimo sa imong sample para mo liquefy sila if after 2 hours wala gyapon siya ni liquefy. Now, our normal liquefaction time kay 30 to 60 minutes. It will happen after the sample has coagulated, then it will liquefy then through. So again, it is important because if dili mo liquefy, dili ta maka-start sa ato ang semen analysis. Now, for its volume and its viscosity, the normal volume of our sample kay 2 to 5 ml. If there is increase in volume, it means that there is prolonged abstinence. So, lapaw na 7 days nga ning abstain kay I can remember sa katong amua pod kay naami nako ikaw ba my classmate nga murag 6 ml or nilapaw to 5 ml ang iyang nga sample, so it means nga grabe ra po kayo to ya pag-abstain. Now if decrease gani ang volume, it means nga improper ang pag-function sa semen nga producing organs or incomplete gyud ang ato ang incomplete ang collection proper. So again, kung incomplete siya kay maka-alter gyud siya sa results. Now, the normal viscosity kay it pours in droplets. It can be drawn into a pipette and form a small discrete droplets and do not appear clumped or stringy. So kung i-dispense gani nimo sa imo nga fluid, diba kay imo mo gina siya i-measure sa graduated cylinder kay para ma-measure nimo ang iya nga volume. Now, if going to check, you will get a pipette and then nimo siya i-allow nga mag-drop. So if makita nimo nga nag-pour siya into droplets, that is normal. Now, if samot ka nang ingani kanang murag dugay kay siya mo bitaw, dugay kay mo bitaw kay um kasi tawag na abnormal siya, abnormal ang dugay mo bitaw. So again, dapat nag-pour siya into droplets. Now for the reporting, we have our grading here. If 0 kay watery and 4 kay gel-like. So 0 watery, 4 gel-like. 0 watery, 4 gel-like. Nigawas na din siya sa board exam. So as early as now, kaya dapat nabalaan na ninyo. Or also po, lahi po nga method of reporting sa viscosity kay low, normal, and high. And if increase ang viscosity, kay mo form siya o droplets longer than 2 cm. And it means that there is incomplete liquefaction. Now, for its pH and specific gravity, the normal pH of our semen is 7.2 to 8, and it is a balance between an acidic fluid of the acid prostate fluid and the alkaline seminal fluid. Now, if there is a increased pH, it means that there is infection or loss of carbon dioxide if kuan siya kanang i-test siya after 1 hour, di ba? Again, ang normal nato nga pag-deliver sa ato nga sample after ejaculation, after collection kay 1 hour. Na if dili siya ma-test within 1 hour, kay there will be loss of CO2. And decreased pH, it means that there is increase in prostatic fluid and ejaculatory duct obstruction, and there is also poorly developed seminal vesicles. Now, for the normal SG, it is 1.010 to 1.030, and it gives a presumptive result of the sperm concentration. For our sperm concentration, so muna na siya ang mga solving apart. Um, the formula or the general formula for our sperm concentration is cells per ml is equal to the cells counted by the dilution factor over area times depth times 1,000 cube over 1 ml. So mura siya kana sa inyong hima, mura nag-solve mo for RBCs and WBC. So ingana na sa diya nga formula. Now for the normal sperm concentration, our normal sperm concentration is greater than or equal to 20 million per ml. Wherein the cells counted is average. So since ang atong ginagamit ani kay kato um, hemocytometer. So, if isa ra ka chamber, kaya na mo kailangan i-average. But if duha ka chamber, mo nang kailangan mo i-average. Then, ang dilution factor nato is 20. Because our dilution is 1 to 20. Now, our area kay depends on the number of squares. Now, we have two methods kung katong 5 nga square sa tunga ang imong gamiton. So, um, lahi ang imong nga area. And also we napoy lahi pod kung ang two large squares ang dira ka mag-count. Now for our depth, kay we have 0.1 mm in the Neubauer counting chamber, and 1 ml is equivalent to 1 centimeter cube. 1 ml is equivalent to 10 cube, and 1,000 UL is equal to 1,000 cube. So that is the formula for our sperm concentration. Now, for our sperm count, we use I mean this is for the still for the sperm concentration. So the procedure is the method that we'll be using is the Weissman method. So first one is following liquefaction of the specimen. We mix the sample thoroughly, collect an aliquot by aspirating the specimen to the zero point 0.5 mark of the WBC Thoma pipette. And after ana kay mo-aspirate ka o chilled water or saline up to the 11 mark and mix. After, discard ka og first 3 to 4 drops as usually do in our when we are using our hemocytometer, and allow it to stand for 2 minutes to immobilize the sperm cells. Kay ko dili na to i-allow nga mo immobilize sila kay lisod kay sila i-count kay nag-movement sila. So muna nga imo siya ipa-stand for 2 minutes to immobilize our sperm cells. And after that, kay you count the sperms in two large squares of the counting chamber. So muna siya atong um, visual grid nga ginagamit. But there is another one katong um, five squares katong sa center I don't know if you can still imagine, pero atong 5 squares sa center. Now, compute for the sperm count using this formula. So, cells per ml is equal to the cells counted times 10 times 20 times 1,000 over 2. But ang short method na kay cells per ml is equal to cells counted times 100,000. Now, if katong gamiton, kay katong 5 squares sa to nga, so ang short method ato niya kay cells counted times 1 million. So again, cells counted times 1 million. So that is for the sperm concentration. Now for the sperm count, kaya ang formula lang gid sa sperm count kay sperm concentration times total volume. So, 'di ba ang unsa man imong ma-result makuha akong sperm concentration, so cells per ml. Now, ang imong total volume kay what is our normal nga volume of our sample? Right. Wrong Dora. 2 to 5 ml. So, for example, cells per ml ay example pila na siya ka um, pila ka cells per ml i-multiply nimo by total volume which is in ml. So ma-cancel out nimo ang duha ka ml. So mo na nga ang mabilin kay number of cells. So a number of sperm cells. So that is for our sperm count. Okay? So that is for the sperm concentration and for the sperm count. For we need to take note that round cells or immature cells and leukocytes are not included in the count. However, ilang presence kay significance and kailangan mo sila i-count separately and kailangan sa nimo sila ma-identify properly because if there are greater than 1 million sorry greater than or equal to 1 million leukocytes per ml, there will be it means that there is an inflammation or infection of the reproductive organs. Now also, if there are greater than or equal to 1 million spermatubes per ml, it means that there is a disruption in the sperm cell production, meaning wala siya ni further into further mature into a sperm cell. So naay ka na disruption sa ato ang spermatogenesis or viral infections or toxic viral infections caused by toxic chemicals and genetic disorders. So those are the things that you need to remember as well. Again, dili sila ginapilog count but they are significant. So muna nga i-count nimo sila separately. Now for our sperm motility, our normal nga sperm motility kay we have two methods of grading. We have kanang 1 to 4, ay 0 to 4 and we have A to D. So ato normals nga sperm motility kay greater than 2 or A and B. So ang motility kay crucial na siya pod for fertility. Kay once ma-present na siya sa cervix kay dapat ang ato ang mga sperm cells kay capable sila nga propel para mo maka-reach sila sa ilang destination sa female nga mga genitals like sa uterus, fallopian tubes and to the egg cell. And it should be assessed using a well-mixed liquefied semen specimen within 1 hour of specimen collection. Again, important jud kayo ang motility. Again, most all of the criteria nga itong mentioned like the vitality, viability, concentration count, anana kay dili ka ingon nga taas man ako number so okay ra ko kanang fertile ako. No, we need to make sure nga tanan nga mga criteria kay normal ka. Kay if one criteria kay dili ka normal, it means nga there is something wrong in you and kailangan gyud ka magpa-check up sa doctor. So kana dapat gyud nga ang ato ang mga sperm cells sa mga males kay kailangan gyud sila nga kanang dili sila weak, ang paspas gyud sila ba, active gyud sila kay para gyud di ba kay kung ingana gani kay ana sila life is a race. Ani nga concept. But in general kay life is not a race. So muna siya, that is for our sperm motility. So, what are our methods of grading? First, what is the method? Our method is cover slip method. So, we place a small drop of liquefied seminal fluid in a pre-warmed glass slide and then place a cover slip. After, evaluate nimo ang motility by scanning 20 high power fields. Dapat i-check nimo ang ilang speed, ang ilang direction. And after ana, i-record nimo ang percentage of sperm nga nag-show og progressive, non-progressive, and no motility. And also we can use also the WHO criteria. So progressive nga motility kay pasabot ana ang sperm kay nag-move linearly or in a large circle. Like nag-move sila in a straight line. Now if non-progressive kay sperm moving pero walay progression. Like nag-move siya pero wala siya ning tapos nag-move siya pero wala siya kanan abot dito ang ana ba. For progressive kay nag-move in a straight line, ana pwede sa diri, pwede ana dito. For immotility or no movement kay no movement. So muna siya ang isa nga method of grading. Now, this is what we usually use, the WHO criteria. So 4, 0 to 4, and we have A to D. So 4, kay rapid, paspas nga pag-move and straight line. Paspas, straight line. And ana side, straight line, straight line, straight line. 3 or B, kay slower nga speed and some lateral movement. Like slower speed and napay mo ato sa kilid nga ana nito. So that is for 3 and B. We have 2 B's di ay. And for 2 and B, kay slow nga forward progression and noticeable nga lateral movement. So hinay siya nga pag-move, hinay siya nga pag-abante, and napoy ka ng mga lateral nga mga movements. Now for 1 and C, kay wala siya ning progress, like wala siya ning abante nga na. And 0 or D kay no movement. So those are our criteria. Ato normal kay 2 up and A to B. So those are our WHO criteria. Now, for our sperm morphology, normal sperm morphology kay 30% kay ang routine nga criteria and 14% kay Kruger's strict criteria. Kruger's strict kay strict siya kay mas dagan kang kailangan nga i-check, mas dagan kang kailangan i-observe. So, we need to take note that the sperm cells that are morphologically incapable of fertilization also results in infertility. Again, kung ang sperm na to kay dili siya morphologically normal, kay mo result na siya into infertility. So, ang sperm na to kay gina-evaluate ang ilang structure sa iyang head, sa iyang midpiece, iyang tail ug sa iyang neckpiece. And important na sila nga hibal-an tungod kay kung naay abnormalities sa iyang head, it means nga dili ka penetrate ang ato ang sperm. Dili siya ka there will be poor ovum penetration. So dili niya ma-penetrate ang egg cell, and if ever na abnormality sa kanang midpiece o siyang tail kay na abnormalities ay naay different na deficiency sa iyang motility. So kung ang head kay poor ovum penetration o kung ang neck piece, mid piece ug tail kay there is abnormality in the motility. Now, what are the normal sperm dimensions? So for our head, for the head sa ato ang sperm, for the head of the sperm rather, kay it is oval and it is 5 UM long and 3 UM wide. And the enzyme containing a chromosomal cap should encompass the approximately one half of the head, and it covers two-thirds of the sperm nucleus. So, muni ang acrosomal cap. Again, acrosomal cap is responsible for the penetration. Muni siya ang part sa head na responsible for the penetration. Now, it encompass one half of the head and two-thirds of the sperm nucleus. Now, ang midpiece, kay this is 7 UM long, and it is the midpiece is the thickest part of the tail kay tungod na siya kanang mitochondrial sheet na dira ang mitochondria kay di ba kung ang sperm na to kailangan mo propel, kailangan siya mo move, kailangan siya og energy, and ang maghatag og energy ana niya kay nanggikan sa mitochondria. So again, mid-piece, 7 UM long. And it is the thickest part of the tail. And our tail is 45 UM long. So those are the dimensions of our normal sperm. So we have here the different na mga what do you call this? Different uh, mga morphologies sa sperm cell. So muna sila ang mga abnormal. So if ever na yung mga abnormal, kay meaning infertile na sila. Okay? So, That is for the normal sperm dimensions. So for the sperm morphology, what we'll be using is right stain. Now, again, what we'll be using for our sperm morphology kay para sa hima, so right stain. So procedure kay mo place ra kag drop of seminal fluid sa isa ka glass slide, maghimo ra kag smear using another slide and imong ipa-dry, imo siya i-mix with methanol, then imo siya i-dip in solution EUC for 10 times. I-dip nimo siya into methylene blue for 1 minute. I-rinse mo with tap water and mo ipa-air dry and mo ipa-examine under OIO. Now, we need to count at least, remember, we need to count at least 200 sperm cells and get the percentage of normal and abnormal sperm morphology. What is our normal sperm morphology? uh, 30% kung routine and 14% kung Ruger's strict criteria. So, ang formula niya, sorry, formula kay normal cells counted over total cells counted times 100. So, muna siya ang ato ang percentage. Again, mga abnormal na to ang mga cells, example, kay kanay mga large, elongated, irregular, amorphous, gamay ang acrosome, naapoy ka ng mga duha ka tail, naay ka ng ingani nga mga, sa muna ang mga abnormal nga mga sperm morphologies. So, we need to count the normal cells and after mo siya divide by 200 times 100, so muna siya to ang percentage. Now, we need to take note that we can also use alternative stains like Jimsa, Shore, or Papa Nicolau stain. Papa Nicolau stain is the stain used in pap smear. So you will know that in our histo class if kato mga na-under na ko. And in Kruger's strict criteria, strict siya tungod kay kailangan na measure ang head, kailangan na measure ang neck, kailangan na measure ang tail size. Also po, kailangan na measure ang chromosome size, ini may evaluate ang presence of vacuoles. Muna nga, um, ang ginagamit na to kay ang routine nga criteria tungod kay dagan kayo nga mga considerations nga kailangan i-take note when we'll be using the Kruger strict criteria. Now, for sperm vitality or viability, or kung mabala na to kung buhi ba, or kanus or unsa mulastlong ang imo nga mga sperm cells. So, the normal sperm vitality is 50% within 1 hour. Decreased sperm vitality may be expected when a specimen has a normal concentration but with decreased motility. Again, kung normal imong concentration pero decrease imong motility, so makakonclude na ka nga mubo gini siya ang ato ang sperm vitality. So, ato ang ginagamit nga method kay Bloom's method. So, same gyapon, place drop of the sample sa glass slide, mag-add kag 2 drops of the eosine. It is 2 times the amount of the seminal fluid and add 4 drops of nigrosine and mix and make an even smear and fix with heat. So, examine mo siya under OIF and again, count at least 200 sperm cells and get the percentage of the live sperm cells. So, ang formula kay viable sperm cells, so viable cells counted over total cells counted, which is 200 times 100. Eosine nigrosine staining method kay ang dead nga spermatozoa kay mo stain siya og red or dark pink, while if buhi kay dili niya wala siya stain, white siya or light pink. So again, here take note kung dead or non-viable nga sperm cell kay i-take up ang color sa eosine under a black background, so mo-appear siya nga red. And if live or buhi siya kay dili siya iyang i-resist ang eosine nga stain, may mo siya nga unstained against a black background. And if there is a presence of large proportion of vital yet dili motile nga mga cells, it may indicate nga defective iyang flagellum. While While kung naay taas nga sini kung taas ang number sa kanang mga emotil or kanang dili mo move and dili pud siya buhi, meaning na nagud siya epididymal pathology. So grabe na gyud kayo ang iya nga sakit ana nga patient. So that is for our the different criterias in our semen analysis or in our semen analysis.